Good afternoon, everyone. Today we shall discuss about the second unit which belongs to me. Actually, it is the third unit of paper 1. The name of the unit is Functional and Comparative Anatomy of Chordates. Functional and Comparative Anatomy of Chordates. In this unit, we shall learn with the knowledge of evolutionary progress of the chordates. So, how the chordates progressed during their evolutionary course with respect to anatomical features of various organisms. We will also learn with the origin, morphology, and even phylogeny of the chordates. At the end of this unit, you will learn or you will understand what do you mean by evolution, how the morphology changes from organism to organism, as well as the phylogeny of chordates. In this unit, the first subunit is a comparative anatomy of chondrocranium, splanchnoscranium, and skull in different vertebrates. So, dear students, we shall study the anatomical features of various chordates in this unit. So, before starting this unit, we must know what is anatomy. Then we will talk about the comparative anatomy on the basis of various body organs or organ systems. So, what do you mean by anatomy? Anatomy is the study of internal structure of organ systems of any organism. Especially, we learn anatomical features in chordates in this unit. So, as you can see in the diagram, they have shown the flow chart of the skeleton. So, once again, come back to the point skeleton. Skeleton is the internal framework of any organism. This skeleton is going to support the organism, it provides shape to the organism, as well as it protects important delicate organs, or we can say vital organs of our body. If we take example of human being, we know that our skeletal system, which is made up of bones, it support our body to walk upright. As we are bipedal organisms, we are walking on two legs. For walking on two legs, our upper portion of the body should be erect, and that is why we have backbone, or we can say vertebral column, and that is totally made up of bones. So it supports your body. Then we have thoracic cage, which help to protect some internal vital organs like lungs, liver, heart, as well as the skull is completely covered by cranial bones. Those are also made up of bones. So these all skeletal structures, bony structures present in any organism are going to support, protect, and shape our body. So the skeleton, which is made up of bones as well as cartilage, right? Some organisms, they make their internal framework made up of bones. Some organisms made their internal framework made up of cartilage, and on that basis we divide organisms into two types. The first one is bony skeletal organisms, and the other one is of course cartilaginous organisms. In this case, or in this flow chart, you can see skeleton is divided basically into two parts. The first one is the exoskeleton, which is present within the integument, so that is very close to the skin, or we can say a superficial surface area which is covered with the bone. And the other one is endoskeleton, that is present deep inside the body. Exoskeleton is further divided into two parts as far as the structure, as far as the content present in it. So if it is a bony exoskeleton which is present from the dermis region, and keratinized exoskeleton which is present in the epidermis. So some organisms are completely or externally covered by scales, which is made up of keratin protein. So their body is externally protected by these scales. They doesn't only rely on internal or endoskeleton, but they are externally covered by keratin protein in the form of skin, in the form of scales, like this. So bony skeleton is present inside, that is into the dermis. Some skeletal structures like hair, some scales which are also originated from dermis region, even our teeth. So they are also part and parcel of the exoskeleton. In the endoskeleton, bones are the main structures. That is why the bony endoskeleton is present in those organisms, like human being, or like other vertebrates which are terrestrial vertebrates or higher organisms. Then cartilaginous endoskeleton, take for example scoliodon, ray fishes, sharks. They internally have cartilaginous skeleton. So their framework is internally completely made up of cartilage, which is elastic in nature, and that is why they can even show flexibility while swimming in the water. And the last one, the part of the endoskeleton is a notochord. It is also a part and parcel of skeleton. Let us come back a little more elaborative part of it. The skeleton is further divided later into cranial skeleton and postcranial skeleton. Cranial skeleton is divided into three. Out of that, two we are going to study. The one of it is chondrocranium, other one is the splanchocranium. The third one is dermatocranium, that we are not going to deal with. On the other side, postcranial skeleton is also there, which we know from their bony structures like axial skeleton, appendicular skeleton. The upper portion of the body made up of axial skeleton, the lower portion of the body made up of appendicular skeleton. In case of axial skeleton, vertebral column is there, notochord is there, even skull region Skull bones are also included in the axial skeleton. Thoracic cage is also included in the axial skeleton. And appendicular, the word itself indicate that appendicular skeleton means appendages. It is concerned with locomotion. So the organisms which have their internal body framework made up of bones which are which are present in the limbs as well as girdles. Limbs like forelimb, that is our hand, and hind limbs, that is our legs, and girdles, that is a pectoral girdle present in the upper half of the body, pelvic girdle present in the hip region of our body. So that is the detailed division of the word skeleton or the skeletal structures present in the body. Out of this, we are going to talk about chondrocranium and splanchnoscranium. Okay, let us see forward. Chondrocranium is also called as endocranium or neurocranium. Endocranium or neurocranium. So it is concerned with the skull bones. It is concerned with the skull bones which are covering the nervous system of our body. And the splanchnoscranium is the visceral skeleton which is present deep inside the body, which also protect our vital organs. Now what about skull? As we are talking about chondrocranium, the skull is present in all higher vertebrates. You can see even in the picture, one of the skull of vertebrate is shown there. The chondrocranium, that includes the box, that is the cranial box, encloses the brain and the capsules surrounding the sense organs which protect the brain. So any injury from external sources, any jerk, any shock from the external sources which may absorb, which may be protected by this chondrocranium. Whereas the splanchnoscranium is the visceral portion of the skull, that is the inner portion of the skull that supports the gills and contributes to the jaws. So those organisms are having gills or gill pouches, pharyngeal gill slits. There also some protection is provided, and that is splanchnoscranium. So we will talk about these two. I repeat, we will talk about chondrocranium, that is concerned with the covering over the brain, and splanchnoscranium is a cranial covering which cover the visceral portion of the skull, especially the gills, and it contributes the jaw also. Let me magnify this diagram to understand this diagram properly. You can see the B point, that is chondrocranium, in which one of the skull is shown, in which some regions are marked. Otico-occipital unit is the first one, and ethmosphenoid unit is the other one. Now, otico-occipital unit is related with the external ear, covering near external ear, and ethmosphenoid is concerned with the nasal septum. From the eyes to nasal septum, this area is covered in the chondrocranium, where you can also find the basipterygoid process present at the base and the nasal opening is shown at the front. Whereas splanchno-cranium is showing the inner or deeply present structures, especially the pharyngeal gill slits or regions of the gill slits where protection is provided. Now let us talk about general embryological development of the skull, how it would have been formed during the course of evolution, during the embryological scenario. Shortly after the initial formation of the embryological central nervous system, the mesenchymal cells surrounding the central nervous system begin to differentiate. That means whenever the basic central nervous system is formed during embryological development, the mesenchymal cells surrounding the CNS begins to differentiate. Slowly, the CNS, that is basically having forebrain, midbrain and hindbrain, that is going to develop or differentiate slowly into some respective part. The outermost layer begins to aggregate into units or centers that forms cartilage. The outermost surface, it is converted into cartilage. In some vertebrates, this cartilage will ossify to form replacement bone. म्हणजे ज्या वेळेला ब्रेन तयार व्हायला सुरुवात होतो एम्ब्रिओलॉजिकल कंडिशनमध्ये, त्याच्या भोवती असणारे जे कव्हर्स असतात ते कव्हर्स ऑसिफाय व्हायला सुरुवात होतात, म्हणजे बोनी स्ट्रक्चर्समध्ये त्यांचं कन्व्हर्जन व्हायला सुरुवात होतं आणि ह्या डेलिकेट स्ट्रक्चरवर प्रोटेक्शन डेव्हलप व्हायला सुरुवात होते. द डेव्हलपिंग कॉन्ड्रोक्रेनियम that forms cartilage at 6 pairs of isolated regions. कुठं तयार होतं बघा. Paracordal region, it is a posterior pair. Polar region, middle pair, only in some vertebrates you will find. Trabeculae, it is present on the anterior side, so anterior pair. The occipital pair, which is present on the back side of the head. The optic and nasal capsule, which is present near ear, eye and nose, where ossification started to develop and cartilage is started to develop. Neurocranium. Now this begins as a cartilage that is partly entirely replaced by bones later on. ॲट द बिगिनिंग व्हेनेवर देर इज अ कार्टिलेजिनस स्टेज, म्हणजे पहिल्यांदा लक्षात घ्या कधी एम्ब्रिओलॉजिकल कंडिशनमध्ये डायरेक्ट बोन्स तयार होत नाहीत, पहिल्यांदा कार्टिलेज तयार होतं. इवन आपल्या हातापायाचे जे बोन्स लाँग बोन्स आहेत ते लाँग बोन्स सुरुवातीला कार्टिलेजिनस फॉर्ममध्ये असतात आणि ते फ्लेक्झिबल असतात की जे अडल्टमध्ये हार्ड होतात, ऑसिफाय होतात, बोनी होतात, पण सुरुवातीला ते कार्टिलेजिनस स्ट्रक्चरमध्ये असतात. म्हणून कार्टिलेजिनस स्टेज जी आहे ती पहिली स्टेज असते. न्यूरोक्रेनियम और दॅट इज अवर कॉन्ड्रोक्रेनियम बिगिन्स ॲज अ पेअर ऑफ पॅराकॉर्डल अँड प्रीकॉर्डल कार्टिलेज बिलो द ब्रेन. ब्रेनच्या खालच्या बाजूला बेसल रिजनमध्ये पहिल्यांदा हे कार्टिलेज डेव्हलप व्हायला सुरुवात होतं. Paracaudal cartilage expand and join along with the notochord from the basal plate. Paracaudal cartilage started to expand and join with the notochord. Notochord बरोबर हे कार्टिलेज जॉईन व्हायला सुरुवात होते आणि एक लॉंग बेसल प्लेट तयार होते. The precaudal cartilage expands and joins to form ethmoid plate. So basically, in the chondrocranium, there are two plates. The first plate is a basal plate, which is developed at the base of the brain, and the ethmoid plate, which is developed on the uh, upper part of the brain region, which is covering the nose, eye, and ear. The cartilage also appears in the form in the olfactory capsule, that is related with the ear, partially surrounding the olfactory epithelium, and the otic capsule surrounds the inner ear and also develop into sclera of the eyeball. So you must understand that three uh, structures or the three organs are going to cover. First, the one at the base of the brain, the second one near the ear, particularly the inner ear, then the sclera of the eyeball, as well as the olfactory capsule. Yeah? So the nose is also covered in this case. You can see in the diagram, this diagram you can once again go through this diagram properly and see exactly how the embryonic development take place in the chondrocranium. The blue color given for cartilage that appears first, but in most vertebrate it is replaced by further as a bone, which become further white in color. The chondrocranium includes these cartilaginous elements that forms the base and back of the skull together with the supportive capsules around sensory organs. Early condensation of the mesenchymal cells differentiate into cartilage, that is shown in the diagram A, that grows and fuses together to produce basic ethmoid, basal, and occipital regions. And in case of B, that latter ossify, that one is latter ossify and forming the basic bones. And sensory capsules. You can watch the diagrams. In case of A, diagram A, it grows and fuses together to produce basic ethmoid, basal, and occipital region. So all blue colored cartilaginous structures are developed first. The first one is shown is a nasal capsule, then trabecula, then polar cartilage, then otic capsule, that is related with the ear, paracaudal region, it is present in the nasal area, occipital region, that is present on the back side, where you can also see in grey color there is a notochord present. So this diagram is showing the developmental phases of chondrocranium. In case of B, you can see that the earlier structures are fused together. Those blue colored structures were scattered earlier. They have not showing their structures proper, but now in case of B diagram, you can see that these structures are closed and they are converted into such a form which can show the ethmoid plate, basal plate, and occipital arch. And in the third diagram, that is in the C, basic bones are formed and sensory capsule is also developed. The blue colored structures are reduced. The reason is the earlier blue color structure which are present in the middle, for example ethmoid, sphenoid, basisphenoid, then the basioccipital, supraoccipital, and exooccipital structures are converted into bones, and that is why it becomes white in color. So, likewise, embryonic development can be seen in the chondrocranium. So, I repeat that chondrocranium, during the development of chondrocranium, what we observe, we study the basal part of the brain, the ear, the nose, and the eye. The bones related with these structures are developed as the chondrocranium. What about a comparative development in other structures, other organisms? In case of cartilaginous fishes, they retain that cartilaginous chondrocranium throughout the life. For example, ray fishes, scoliodon. These fishes have retained, continue to develop into adults also as a cartilage only. So these fishes are cartilaginous fishes throughout the life. In case of bony fishes, lungfishes, and most of the ganoids, they retain highly cartilaginous neurocranium that is covered by the membrane bone. So internally the bones are not developed, but externally cartilage is remain in this case. Other bony vertebrates, in the other bony vertebrates, embryonic cartilaginous neurocranium, that is chondrocranium, is largely replaced by replacement bone. The process of endochondrial ossification occurs almost simultaneously at several ossification centers. You must know what is the meaning of ossification. Ossification means bone development, conversion of what is that? Conversion of your cartilage into bones is the ossification take place. So in all the bony vertebrates, maximum cartilaginous structures which are present in the embryonic part, they are converted into bones. Then neurocranial ossification centers. For example, the first center is occipital center. That cartilage surrounding the foramen magnum may be replaced as many as 4 bones: basioccipital, exooccipital, which are 2 in number, and supraoccipital. So basioccipital is 1 in number and supraoccipital is also 1 in number. So these bones are converted from cartilage to bones in the adults. So what is the foramen magnum? Foramen magnum is the cavity present at the base of the skull through which the spinal cord is running downwards. In case of mammals, all 4 occipital elements typically fuse to form a single occipital bone. If you remember the earlier studies of your skeletal system, the cranial bones are of various types. One of the cranial bone is occipital bone, which is present on the back side of our head. So these 4 bones, basioccipital, exooccipital, and supraoccipital, these 4 bones are joined in case of mammals to convert into a single bone that is called as occipital bone. But in case of tetrapods, those are walking on 4 legs, especially apart from the human being, as well as apart from the mammals, they articulate within the first vertebra via the 1 or 2 occipital condyles. 1 in case of reptiles and birds, whereas 2 in case of amphibians and mammals. So these occipital condyles are formed, which is developed in these skeleton skull region or the skull bones. Here you can see this occipital bone in the inferior view. There is a large cavity present between, in the middle, that is the foramen magnum, through which the spinal cord is running downwards from the brain through the vertebral column up to our lower part of the back. There are various regions present in the occipital bone, and these regions further attached with each other, connected with each other, and they are forming occipital bone. So earlier it was also cartilage, but later on it is ossified into bone and becomes a protected structure. The second one is a sphenoid center. In case of sphenoid center, in case of sphenoid region, basi-sphenoid bone, that is anterior to basi-occipital region. बेसी ऑसिपिटलच्या अँटिरियर म्हणजे वरच्या बाजूला हे बोन प्रेझेंट आहे. प्रीस्पिनॉइड बोन दिस इज द सेकंड वन. साइड वॉल्स अबाव द बेसिस्पिनॉइड अँड प्रीस्पिनॉइड विच फॉर्म्स ऑर्बिटोस्पिनॉइड, प्ल्युरोस्पिनॉइड अँड ॲलिस्पिनॉइड. So these all regions contribute to form a sphenoid bone. You can see in the diagram the second one, sphenoid. This structure is indicating which is present near the eye that covers the orbit also. Even you can also see the other bony structures which contribute the chondrocranium. The first one is ethmoid, which is present in the nasal region, the sphenoid which covering the eye, the otic region which covering the ear, and the occipital region or occipital bone present on the back side of the head. And still you can also find a cavity that is foramen magnum at the center. The third one is ethmoid centers. It tend to remain cartilaginous to form, and the form is remain throughout the life also. Anterior to sphenoid. What is the location of the ethmoid center? It is anterior to sphenoid. Let me show you the earlier diagram where you can see the ethmoid center. The ethmoid one, which we can see at the top, which is red in color, and in the region of nose. What does it indicate? Our tip of the nose is still moving structure. Whenever we move, it can move, and it is flexible, and it gives shape to the nose. That is why it is a cartilaginous one, and that is remain throughout the life as it is. This is a lateral view of the ethmoid center. The ethmoid region is clearly visible within the bisected skull present in the upper diagram. In most of the mammals, the nasal chamber is large, filled with ridges from the ethmoid bones. That's called the turbinals or ethmoturbinals. These bones are covered with olfactory epithelium in life and serve to increase the surface area for olfaction, that is, more acute sense of smell. Another ethmoid bone, the cribriform plate, separates the nasal chamber from the brain cavity within the skull. आपल्या नेझल रिजनमध्ये जी पूर्ण नेझल कॅव्हिटी असते, this nasal cavity is separated by this ethmoid bone, and it is externally covered by the cartilage. The fourth one is the otic centers. The cartilaginous otic capsule is replaced in lower vertebrates by several bones: prootic, opisthotic, epiotic. One or more of these may unite with adjacent replacement or membrane bones. In case of frog and most reptiles, you can see opisthotics fuse with exooccipitals. Birds and mammals, prootic, opisthotic, and epiotic unite to form a single petrosal bone. The petrosal in turn sometimes fuses with scamosal to form the temporal bone. The developing splanchnolocranium is also characterized by pair of cartilaginous aggregates that form pharyngeal arches. It is derived from neural crest cells. The neural crest cells migrate into the walls of the pharynx to form the pharyngeal arches between the gills. The anterior branchial arches that supports the mouth are the jaws. The mandibular arches becomes divided into dorsal and ventral components. The dorsal components called as quadrate cartilage or palato pterygoquadrate bar, and the ventral component is called Meckel's cartilage, in which hyoid arch and branchial arches are involved. So, in this lecture, we have learned about the chondrocranium, in which four different components we have learned. The one, otic centers; the second, ethmoid centers; the third, sphenoid centers; and the one is the occipital centers, because all these bones are going to develop into chondrocranium, which covers the brain of the vertebrate. So, in the next lecture, we shall study the next part, that is planchnochondrium. Till then, goodbye.